It's another Tuesday here, noon. We have another seminar in our bioinformatics genomics series, and very nice presentation last week. Today we have Rachid Karam, and he will be introduced shortly by Alice Lee from Boston Children's. Just a reminder, next week, May 26th, we have Tomasz Nowakovski, who will be talking about single cell epigenomics atlas of the developing human brain and organoids. So that's next Tuesday, same time, same place. I wanted to highlight that this Friday at 2 p.m., we have the last of our 4 sessions of the Genomics Symposium at Boston Children's. We had a very lively panel discussions over the last 2 weeks. This Friday, we have Ann Poduri, our own clinician and researcher, giving the final closing keynote. So that's at 2 p.m. on Friday. And if you guys wanted to capture, I'm going to everyone and send the Zoom link to that meeting. So if you wanted to mark your calendar quickly for this Friday, 2 p.m., we'll have the seminar by her on epilepsy and how she's using genomic data in her research. Okay, so couple of organizational remarks. We will have a question session at the end of our presentation today, and we allow people to unmute. You will be able to unmute, and if you want, turn on your video as well and ask the question directly to the speaker. If you want to remain shy, you can type it in, and Alice is going to read it on and send announce in the forum. Okay, so 12:02, we have 45, almost 50 participants right now. Alice, I think I'll pass it to you right now to introduce the speaker, and we can fire up. We can get started. Thank you everybody for joining again today, and looking forward to the great presentation. And Alice is muted, so we cannot hear her, but she should unmute.
Hear me?
Yeah, we can hear now. Thank you.
Yeah, I'm happy to introduce today's speaker, Dr. Rachid Karam, to you. So he's a medical doctor and also PhD of oncogenetics, and he's a director of R&D, translational and clinical research in Ambry Genetics in California. And he also serves as a chair of ClinGen CDH1 expert panel. And he's going to talk about his published work on splicing profile by capture RNA-seq identifies pathogenic germline variants in tumor suppressor genes. And also he's going to briefly touch on some of his unpublished data as well. We are looking forward to it. Okay, Dr. Karam.
Yes, first of all, thank you for having me to discuss this paper. So this paper was just recently published, February of this year. The main author, he was my postdoc at the time, Landreth Tyler. Now he is a scientist at Ambry. Okay, so this is the paper. We had a lot of collaborators there, a lot of clinical collaborators, because the main goal here, I mean, you are all aware, I'm sure, like we didn't created RNA sequencing, right? RNA sequencing is a technique that has been broadly used. What we really did here is to implement this test clinically with the first, paired with the DNA testing for patients with hereditary cancer. So as you may know, traditionally genetic testing was DNA based, right? You test the DNA and you see if the patient has a pathogenic mutation. What we did is we added to the DNA sequencing component, we add RNA sequencing to see, to measure the impact on the VUS rates and to impact the variance of a non-significance variance that we cannot properly classify, and to measure the impact on detection of new variants, right? Because our DNA sequence has its limitations, and I will elaborate a little bit further. So it was a multi-institutional collaboration, which was a very rewarding process. And I also want to say, like I mentioned, we were not the first to use this technology, RNA sequencing, to show that it can improve the diagnostic yield overall with people with many different diseases. This is coming comes from the Broad Institute. They were, in 2017, they show they were able to improve genetic diagnosis in Mendelian diseases with whole transcriptome sequencing. And in this project, they focus a lot on the, I think, muscular dystrophy, and they actually analyzed biopsies to do the whole transcriptome sequencing approach. This is also a very interesting paper that was published last year from Steve Montgomery's lab in Stanford with the Undiagnosed Disease Network. And what I love about this paper is that one thing, they use blood. And they did whole transcriptome sequencing in a relatively large cohort, 100 people, that have done previously exome sequencing in the DNA level, and they come back negative. So they rerun retrospectively, right, the RNA sequencing, and they were able to show a 7.5% increase in the diagnostic rate, which means like positive cases, right? It doesn't come VUSs and lower, and addiction of 16.7% with improved candidate gene resolution. Right, so this is part of the background to give you like an idea that this has been used in the research setting, especially. I talked with Stephen and took them for this 100 patients 3 years, right? So I'm quite sure clinically that will be a hard test to implement if you tell your the medical provider or the patient that you have to wait 3 years to get the results. So that was in part what we were trying to do, and we did, to streamline this process and to have this delivered to more people. And this came from this previous knowledge. My postdoc was mostly, it was in RNA, it was in an RNA lab. When I went to Ambry in 2014, I really thought, was like, well, there's all these VUSs that I'm seeing here. And if you had like, if you could do RNA analysis, which we would do normally in the clinical lab, you would be able to elucidate a lot of them, right? Because I'm not sure how, if you understand well how the variant classification system works, is based on the American College of Medical Genetics and Genomic Guidelines, ACMG. And you use different pieces of evidence to different criteria to reach a final classification. And RNA data is one of those criteria. It's a strong criteria. If you have RNA and data, you can add to the clinical data, you can add to the population frequency data we get from NOMAD, you can add for the computational prediction tools, because we do in silico predictions. You can add that RNA data, and because it's actually wet data, you can apply strong evidence to it. It's a criteria called PS3. So what we did here, when I joined Ambry, the goal was, okay, let's look at a cohort of patients that have VUSs that are predicted to affect splicing, and let's see how, let's do the RNA assay retrospectively, right? So we invited these people, look, you tested with us, we find this positive VUS, it's predicted to affect splicing, we would like to offer you RNA testing to see if we can improve the classification. Because VUSs are tricky, right? I'm not sure, there are people that may not be clinicians here. So usually the classifications, you have like a 5-tier classification system. Following the ACMG, we start with benign, likely benign, and VUSs, which are not clinically actionable. And then you have the likely pathogenic and the pathogenic variants, which are clinically actionable, right? People that have, for example, pathogenic or a likely pathogenic variant in gene CDH1, I did my PhD on CDH1, the recommendations are that these people do risk reducing or prophylactic gastrectomies. So they remove their stomach to prevent the develop a very deadly disease called diffuse gastric cancer. So just for you to understand how important the pathogenic and likely pathogenic, the repercussions of classifying something as pathogenic and likely pathogenic, they are what we call clinically actionable variants. If there are guidelines, your physicians and genetic counselors, they recommend those implementation of those guidelines. So we showed here in this paper retrospectively that yes, if you look at the two first bars, before you perform RNA genetic testing, a significant proportion of the cases were classified as a non-significance VUS. And after you post RNA genetic testing, you can see that a significant proportion of them got reclassified. And what I think is very important is more than half of them ended up being classified as clinically actionable, likely pathogenic or pathogenic variant. So these are the groups, these were the people that were not getting the medical attention that they need before. And now because we just add RNA data with other lines of evidence, we reach the full classification for this variant. This paper, it's open access, and if you are more interested in variant classification, the supplementary material has like the details of all clinical criteria from ACMG based that we used to reclassify those variants. Wasn't only RNA. But all these assays, these studies that I showed you, the Bro, Stevens, and ours too, they were all research protocols, and they were all retrospective in nature, right? And by retrospective, I mean you do the DNA testing, and the DNA testing is negative, and then you report that back to the medical provider and to the patient. And then you tell them, look, you may benefit of doing RNA testing. And then they would send us another sample, and then we would do RNA testing as a research protocol. And that has limitations because, like our research lab, it's part of R&D, so we're not part of the clinical lab. So it's a much lower throughput, and it's no clinical TAT. I used to joke when people, what is the TAT? Is a clinical, it's an academic TAT. I used to say, because it could take months, right? You have postdocs working on this instead of having like a large clinical lab like we have with robots. Everything is automated and much faster, right? And clinically, I think was very important. 90% of we invited 1,000 people to participate in the study, and this was for free. All you had to do was go back to the counselor and get another blood draw. 90% of the patients, they were lost on follow-up. Only 100, less than 100, not 93, show up and actually send additional blood samples. So we really realized that the moment to get the RNA sample, clinically, if we believe that this can really help improve the diagnosis overall, the positive yield of genetic testing, which is relatively low, by the way, right? And I'm not sure if you know, but approximately only 10 to 15, 20% of the cases that are tested for cancer, for hereditary cancer, test back positive with pathogenic or a likely pathogenic variant. So, and that's because there are limitations as well with genetic testing. And one of them is that this approach, DNA only, or even the retrospective, it's not ideal to identify unknown deep intronic variants that may be pathogenic. Like, right, like all you know, the introns are huge, they are poorly conserved, there is a lot of variability. Yeah, you could do whole genome sequencing, but you ended up like with hundreds of USs. And the advantage of doing like whole transcriptome sequencing is that you can detect abnormal transcripts and then go find the abnormal variant, right? So that's the approach that we are thinking. So the idea is here, pretty much like you may know, the clinical testing, when we do the multi-gene panel testing, we select like about 85 genes that are associated with hereditary cancer, and how the test works. It's a probe-based system. It's NGS, next generation sequencing, and you have the captures, and then you capture the exonic and the boundaries of the introns only, and that's the data you have for DNA analysis. So you will identify all the variants there, and you may identify splicing VUSs in this region here, and then you can do retrospective assay. But the problem is the way the test was being done, all these regulatory regions that are located inside the introns, they were being missed. And our hypothesis was that we would be able to detect variants by doing RNA sequencing that we would not detect if we were doing the DNA approach only. So that was an intro. Now to this what is described in the paper too, and then the whole idea, the whole concept here was like, okay, the patient goes see the genetic counselor, then it's a blood test, and then that's the time to the kit, instead of having one EDT tube for DNA, you have a packaging tube for RNA. So the packaging tube stabilizes the RNA at room temperature so they can ship that test to us. And then we will do both assays in parallel. We are doing the DNA sequencing in parallel, and then we're doing the RNA sequencing in parallel. And I think this is an important part here to make a distinguish different approaches, because people use the term RNA sequencing. It's a very broad term. There are many types of RNA sequencing you can do. Whole transcriptome is the one that I was describing on the Bro and Stephen's paper too. But we decided to do a different approach. We decided to do capture RNA sequencing that was also published before. So pretty much we're using probes to capture the genes that we care the most. Because like we really, if you want to automate this to make this clinically available to most people, why would you sequence the entire transcriptome if you are really interested in actually studying some more well-characterized genes like tumor suppressor genes that if found to be mutated in germline may cause hereditary cancer, right? So the goal was like, let's focus on 18 tumor suppressor genes that are ubiquitously expressed in blood. They usually have a major transcript isoform. Two, they are clinically relevant. So we focused on those 18 tumor suppressor genes, and we are capturing the mRNA from them, and then we were doing alternative splicing detection. And in the end, our analysts merge all the data from the DNA with the data with the RNA. And so that's the paper that we're going to discuss. That was part of the supplementary figure, but it explains how the... One of the challenges that we have, because when we had to build our own, excuse me, we had to build our own reference database, because we didn't want to be comparing with databases that were available elsewhere, because who knows how they were doing the experiments, right? So we decided to build our own control database, so we would not be comparing oranges to apples, right? So we use pretty much like the same protocol that we validated to do clinically for the patients. We validated to do, we run on our control data set. So this is the data for the first 350 controls. Now we have thousands of them. Here what we did, we tried to build a control database that was as diverse as possible, right? So you can see here different genders, different age range. We also focus on different ethnic groups. But then one interesting thing that we found, it's in D here on this violin plots, you can see that we actually don't see much difference with regards to any of those factors. But here specifically, ethnicity, it doesn't seem to have like any major difference. What we did observe is, and this is something that we knew, is that everybody has alternative splicing, right? And we used to joke, like if you sequence someone and you don't find alternatives, that's when you call the doctor, look, this patient has no alternative splicing. So like, but yes, pretty much, and that's important. That's important because when you look at patients and you find a transcript, you want to know, is this an abnormal transcript or is this an alternative spliced transcript, right? Because there is a distinction. You need to distinguish between alternative versus abnormal splicing. And that's why we had to build this control data set. And then here in E, we have the hierarchy cluster analysis, HCA, and that was also to see if there was any, and we didn't see any association with any of these factors either, right? Not only ethnic group, but also gender or age, which was actually good to see, because I think that reduced a little bit the complexity. One thing that I mentioned is, and then I move into figure 2 of the paper, and this is just part of figure 2. Figure 2 is huge, so I'm just going to focus on 2 genes, BRCA1, BRCA2. But you can see here that overall we detect splicing in pretty much like every gene in blood, alternative splicing, right? So these are the normal controls. These are not patients. And you can see here that we use very stringent, we use a PSI. PSI, we didn't invent it, it's a bioinformatic calculation to tell you the percentage of splicing events that you have. So PSI above 5%. With a coverage above 50x, that was our quality control for this, right? So these were and present in more than 5% of the control cohort too. You can see that a lot of those genes here, you have different splicing events, some genes more than others. And as we knew already, BRCA1, it's a very gene that has a lot of splicing, BRCA2 too as well. UTY8. So here we have data for all these genes, either in the main figures or the supplementary figures of the paper. But here I want to focus on the BRCA1, BRCA2. You can see that you have all those transcripts that are out of frame, the frame shifts, right, that are supposed to be degraded by nonsense mediated decay. So these ones here, you see them at a lower PSI, which is not surprising, right, and less frequent in terms of how many patients do you see them. Because this could be like trash splicing that didn't happen properly. This could be like some sort of regulatory element, like a way to keep BRCA1 down, right? You just produce those transcripts that are degraded by NMD. But then we separate them by in-frame too. So in-frame is different. So in-frame, you don't lose the frame, so you don't have nonsense mediated decay, right? So those transcripts are predicted to produce proteins. So they could have a biological function. This is something that we didn't investigate here. And there are others that are doing. There is some very well-known alternative splicing events. Here it was good that we detected splicing events that others had described before. There is one here that you have like a huge skipping of the linker region of the BRCA1 gene that is frequent in a lot of people at different levels, some as high as like 25%, that we don't know exactly what it does. And then here you see BRCA1, just a comparison, is not as splice prone as BRCA2 is not as problematic or has as much splice as BRCA1. And this is something that we learned. These are all the 18 genes that we did, the capture RNA sequencing, right? So you can see you have like polyposis genes like APC, predisposed to colorectal cancer, ATM, BRCA1, BRCA2, the Lynch syndrome genes as well, the P53. All this data is available in more detail in the paper. But what we saw gene-specific difference, right? So some genes, RNA is definitely having a much bigger impact that is having in others. So this paper was pretty much the validation of the test. The test was launched clinically. MGH was one of the pilot sites, so they started doing the test in the beginning of 2019. But then the test was made clinically. Was made clinically on September. That means that the test was available for patients to order, and it was approved in New York in December. So it's officially available for everyone in the country. This is something important to say. All this validation work here, we already migrate from the R&D environment to a clinical environment, right? We were validating the test clinically. So that's a big shift. So now you're going from our research lab to our like super lab, which is like automated and has like 24/7 resources and all that. So that was part of the goal of having this test delivered with the same turnaround time as we have the DNA test today. So have them done paired, right? So what we call the DNA plus RNA genetic testing approach, the paired approach, right? So what we did to validate the test clinically, we selected a bunch of variants that were previously before the study classified as pathogenic and likely pathogenic to see how they perform with this test. And then there are many ways you can show RNA sequencing. I'm sure you know that. Sashimi plot, this is one way to display. I like to see sashimi plots because it gives you a more quantitative and a qualitative view of the transcript. So here briefly, I always ask our bioinformaticians to please color the patient in tuna, and then the controls in salmon here. So you can see the patient here. You can see with the sashimi plots, right, like the peaks align, these are reads aligning to a specific axon. And then the lines, the number of splicing events connecting this axon to this axon here. So like normal splicing. And you can see in this patient that we see splicing happening somewhere in the intron, right? That shouldn't have spliced there. And then the advantage is now we have the chromosome location, so we can go there with Sanger analysis, for example, or a DNA test, and look specifically for a variant, and we found this variant here. This variant is a well-published variant. There are patients with ataxia telangiectasia with this variant. So we use this variant as part of the validation. I don't think we include this data to the paper because one of the goal of this test was to detect this type of variants, what we call deep intronics, right? So very far away from the plus minus 5 boundaries of the axons, right? So here we talk about a minus 1050 A2G variant that results in this out of frame pseudo-axon inclusion, right, which leads to nonsense mediated decay. So this is how you read this, in case you didn't know how. This was a sample that we use in the validation. We will be using a lot of sashimi plots, and the paper has a lot of sashimi plots too, so just we are on the same page. So we validated all this data looking using our orthogonal method. We call this technique clone-seq. We published about 3 years ago. It was pretty much we would just do like a target approach with primers, like a RT-PCR clone, and then sequence everything. So it's our orthogonal method is different from RNA from capture to show here like the positive versus the health control for all the variants. So here are just the variants we are showing in the main paper, but you have sashimi plots for everything there in the supplementary figures too here. In the supplementary figure too there. How are we doing with time? 28 minutes. So the next step, and that's figure 4 of the paper, was to so we validated the test. So that was we did that was part of the test validation, which is required for you to launch a test clinically, like this test is, right? And then we start testing people from the pilot sites, people that we were collaborating with, to see if our hypothesis that this would lead to less VUSs and more positive cases was true. So the first 1,000 consecutive clinical cases, we did see a 9.1% in the positive yield. So a relative increase in the positive yield, which was significant, and we also saw a reduction of about 5% in VUS rates. Just on the first 1,000, we know the first 1,000. The goal is the more the better, and that's the data, published data that I have to share. This is the tip of the iceberg, right? We just recently run a prediction. How many variants we have already tested 40,000 variants. How many variants we have seen of the ones that we are predicted that we could see? We have seen less than 4% of them so far, even with 40,000 people tested. So that's why we think this is part of genetic testing long term, because there is a lot of things to be discovered there and for us to learn. So this is figure 4. Overview figure 4. These are the cases that would have been either negatives or VUSs of a non-significance if we had done the original approach, a DNA only approach. Right, so I'm going to discuss in a few more details some of those cases, because I think they are very emblematic here. This is the proband is a female with mixed Caucasian and Asian descent. She was diagnosed with ductal breast cancer at age 25 and 33, and then ovarian cancer at 44. Right, and I think this is a very emblematic case because the lab that classified this variant before classified this variant as a VUS, because had this variant being classified pathogenic, ovarian, this was a totally preventable cancer. ovarian cancer, right? People can, if they had a positive, you can recommend surgery if you have a VUS, if you have a positive. These people have access also to treatment targeted therapy, such as PARP inhibitors, for example. But you can see here how the RNA data helped us shift the classification. So you can see in the sashimi plot, again, the patient has this abnormal transcript, this R dot here. So insertion of 8 nucleotides, right? That was not found in any of our controls. And what was interesting, and then when we look at the DNA, we saw this variant that was creating a new acceptor site, 8 nucleotides upstream. So the variant was pretty much creating a new acceptor site that the cell was using. And this was the evidence that we used as part of the criteria to classify this alteration as pathogenic. So this was an interesting case that it took for the patient a long time to have finally a conclusive answer for her test. And here is just to compare, like, when another way to display the data, right? Like if you compare, remember the question, we always compare to our control database, because we want to see, is this alternative splicing found in other people, or is this something like, no, nobody is expressing this transcript, this R dot with this insertion of, none of our controls are expressing it. So just the patient is expressing it, the patient with the variant, with the -9 variant. The other example is also of BRCA1, pathogenic, is a +6. We are suspecting this is a Ashkenazi Jewish founder mutation, because we have seen since in 3 independent, I don't know, we have to do haplotype. It's a little preliminary to say, but we are seeing this variant, and it definitely has a clear effect. It causes exon skipping. So that other variant was insertion of a little bit of the intron out of frame. This one, it's skipping of the exon, right? So what you can see here on the sashimi plots, you can see the exon, the normal folks go here to this exon, to this exon, this exon, to this exon. Then you can see that in the patient, you have this R dot. So you have splicing skipping this exon here, right? And you, same thing as we showed you before, none of the controls expressed this abnormal RNA. So we use this as evidence to classify the variant. So 30 minutes, I'll just briefly go through unpublished data. I just thought we would be interesting because we just recently finished analyzing the first 10,000 consecutive patients sent for clinical testing. So 17 pilot sites, the same approach, clinical testing, done the clinical lab, everything's clinical. The clinical research team, my team, we just do the analysis of the data right now. The clinical reporting team does the variant assessment, so it's all independent, right? We're just looking at the results from everything and putting it together for those 18 tumor suppressor genes to evaluate the positive yield, so the cases that are classified as pathogenic, clinically actual pathogenic, like pathogenic, or the VUS gene, the improvement or not in the VUS, in these 18 tumor suppressor genes. Then you can see here for the first 10,000 consecutive patients that yes, we are seeing a big impact, and some genes more than others. ATM, we are finding a lot of deep intronic variants. It's really interesting, I find, because ATM is huge, 63 exons, is like an F1, and we really find some really cool, interesting variants that were not detected before at all, just by looking at sashimi plots. And then confirming them at the DNA level, of course, and doing variant assessment. But you can see ATM is the one that has benefited the most with regards to new pathogenic, but BRCA1 as well, and MSH2, a Lynch syndrome gene, predisposition to colorectal cancer, right? Also impacted, and then VUS rate, so how decreasing number of VUSs also for different genes here. And then just one case example, because the first 1,000, we really didn't have like a real deep intronic. I really want a deep intronic. And then like the first like 1,200 cases, we found a dipintronic. And then we start to see them more and more often. And I will share one example, just because this was something that we really wanted to do in the design of this test. It's kind of like shift a little bit the paradigm. Instead of using DNA to detect the RNA, we want to use RNA to help us look where, to tell us where to look in the DNA, in the intron specifically, to find a variant there, right? That was our hypothesis, and then it was good. The more people we start to test, we start to find these variants. And this is one example, a novel variant never before described, plus 829 inside the intron, right? So well beyond the range of any multi-gene panel testing. And this was also a family that has been tested and it struggled with only having a clinical diagnosis. So for FAP, it's a very severe disease, right? People have multiple polyps with almost 100% penetrance in some families of colorectal cancer if you don't do colectomy. And you can see our proband had polyps at 33. His dad had clinical diagnosis of FAP with a colectomy performed at 55, even without the genetic diagnosis. It makes sense. But they had a strong family history of polyposis. So we should run the clinical test, and then what we saw that pathognomonic sign for something dipintronic. You can see the sashimi plot of the patient was telling, okay, you have the normal splicing here, connecting exon 8 to exon 8 to exon 9, and you see the huge intron in between here. But then you also have reads connecting somewhere in the middle of the intron, and then that told us something is going on there. So we have the genomic location. We go there, we sequence, and we found the variant. And then we find the variant. Then what we do a full variant assessment, right? Follow ACMG. We look at the IGV to see if these are bidirectional, real RNA reads. We do manual inspection. Then we look to see if the sequence actually is doing something, because is it creating a new splice site? And in this case was creating, and then it's a qualitative test. Input, it gives you the R dot here, pretty much like of what we have found. It was an insertion of 73 base pairs, out of frame transcript, therefore targeted for nonsense mediated decay. And then knowing the genomic coordinate, then it's easy. We can do a Sanger analysis, and then we can confirm the variant by doing Sanger, for example. And that's how we identify the variant. Then we run in silico predictions on everything. Here is just to show that this is, you can see by eye, but your predictions are also showing creation of a strong splice site, right? Which supports the activation of that cryptic exon there. Suddenly the cell detects as if it was an axon and inserts that portion of the intron. Very interesting to the mRNA. And then we test more people. Oh, I didn't update this slide. We also test the grandma. She's also positive, and she had she did colectomy too. So we tested. Then knowing where the variant is. We tested the father, and sure he was positive, and then we test the grandmother, and she was positive as well for the variant. And these are just, like I said, we don't use RNA as a single line of evidence. We put this in context of ACMG guidelines, so we need to fill a threshold. And then all the RNA data is used as strong evidence, but we also had multiple programs, which gives us a clinical criteria, which is called PS4, the in silico criteria, which is a supporting evidence because it's not as strong as web data, right? So it's a supporting PP3, and then rarity, we look at NOMAD. This was not found in NOMAD, which gives us a moderate criteria PM2. And that's sufficient to call the variant likely pathogenic, which is clinically actionable, right? Now, and these are our collaborators, internal and external. And I also like to thank you for your time. And if you have any questions, I'll be happy to answer.
Thank you, Dr. Kemba. Are there any questions? So I'm actually curious, when you identify likely pathogenic or aberrant transcript from RNA-seq. If you go
Mhm.
back to the DNA-seq, then what fraction of those aberrant transcripts actually can be linked to DNA variants?
That's a very good question. I don't know the number exactly, but that's why we have analysts to make the association, because we see alternative splicing variation. But if we don't have a DNA to report, we are not reporting only RNA data, because the clinicians would have no clue what to do with it at this point, right? So this is a clinical test, and this was a feedback that we got from the clinicians was, no, we want a clinical, the DNA variant, and then that's what we are reporting, the DNA alteration. If we can find a DNA explanation for some alternative splice event, could it be environment? It could be so many things. That's why I say this is the tip of the iceberg. Whenever we have the entire data, the set analyzed, I think we will uncover a lot of cool stuff.
So, very quick question. So the RNA is just standard RNA, right? So you start with whatever everybody else starts. An RNA.
We do both in parallel.
Yeah, but okay.
With regards to workflow, we do both in parallel. Like they just run in parallel, and then the pipeline merges the results in the end.
But the way you are extracting the RNA is just standard methodology. You don't do anything special.
We do, it's automated. We have an automated protocol for RNA extraction.
Okay.
I think this is capture RNA-seq, not a regular RNA-seq, right?
It's capture. Yeah. So you have the probes to hybridize, and you're just capturing what really. But we do. It's described in the paper. We do the ribosomal depletion as well. But yeah, the probes are what capture the RNAs that get sequenced. Yeah, which really improves our detection range, because if you see BRCA1, BRCA2 in whole transcriptome, like you have 10 reads, and several regions are not even covered, and then we could use whole transcriptome even if we want, because the quality is so low for BRCA1, BRCA2. So we had to really, let's prioritize the genes that we want.
Yeah, so that would have been my next question. That what coverage would you need from a standard RNA-seq to at least see a few of those splice variants you find with your capture? I'm just curious.
By standard RNA-seq, you mean
Yeah,
like transcriptome sequencing?
Yeah.
Yeah, good question. Our quality control is 50x. So anything that is under 50x, I don't even trust.
Okay.
Yeah.
So, 150 and 200 is also pretty standard. So did you ever run any comparison? I'm just curious.
For what? Sorry?
So if you start with 150 or 200x, did you run any comparisons, whether even just an artificial comparison, just subsampling, whether you could see some of those, let's say, BRCA1 splice variants that you
yeah, you could. I think the ones that I show here, they work with coverage like of 200, 300x. Yeah.
Okay.
These are the lower genes. ATM has ton of RNA. APC has ton of RNA. ABRC1, BRCA2, or that's where really the capture made the difference.
Okay. I mean, absolutely. So your method is going to be definitely biased against genes that are not expressed peripherally. So although I think BRCA is expressed in immune cells, so I would expect some expression, but those genes that are not expressed in the peripheral blood, obviously you're not going to see those.
Yeah, and that was the surprising part from Stephen Montgomery's paper, because if you look at his paper that he published in Nature Medicine, I was like, we do for cardio and research and some neurogenes, but he systematically looked at a lot of genes that involve intellectual disability, and he was able to detect developmental disorders in blood. So I thought that was an interesting approach that he's showing blood that even though they are not tumor suppressor or genes that are ubiquitously expressed, the technique still yields some increase in the positive yield.
Okay, thank you.
That's a really cool paper. I recommend people read if you haven't yet.
Yeah, thank you.
Well, let me unmute Nick. You had a question.
Yeah. In terms of the statistical analysis, I guess I'm wondering how this experiment compares to kind of a more traditional whole transcriptome sequencing, and particularly with whole transcriptome RNA-seq, normalizing the data is kind of one of the most important parts of actually identifying the variants. It just seems like this experiment would have a different normalization process. I'm just wondering if you can comment on that.
Well, that's the entire figure 1 and 2. We pretty much had to build our own control database because we didn't want to go and use the published data set as a control because we don't know it. So we want our control to be as similar as the cases as possible. So we just pretty much run the same thing that we run to the patients, we run to the controls, so we can have more confidence that what we're comparing, it's comparable. And we have a statistics team, so the variants need to be statistically significant different from, and they need to pass a threshold that was pre-calculated from what is known, because not every case is as clear as this. There's also the case, like we are saying, very problematic because there is that alternative splice region. So what do we do if variants in that region? Like we see if there is abnormal, we see there is alternative splicing. We see the patients with variants affecting those axons have more alternative splicing, but we don't know what is the consequence clinically. So there is a lot of studies to be done to really on a gene by base.
Yeah, we have a couple more questions. So maybe Liang.
Yes.
Yes. You can turn on your video so we can see it.
Right. So I actually have a follow-up question because I think I have the same question which the first person already asked. Because for the alternative splicing, this kind of project, usually we need more coverage of RNA-seq data to identify this novel splicing, site also. So I was just wondering, do you actually consider except the coverage of RNA-seq data, do you also consider about the sequence length, say whether it's 100 pair end reads or 150 pair end reads, or the sequence length will affect the accuracy of identify this kind of alternative splicing?
The sequence, the bigger the better, right? But we do pair 250, and that's working for us because all you need to detect is actually that junction with the cryptic. So you see that RNA going from one side to one side. So the bigger the better, but paired to 150 works well when we are detecting the dipintronics for those 18 genes, right? That's the payoff, and that's why we're doing this clinically too. Research may be more challenging because you may want to look at everything, and that's the payoff here. We get better coverage because we're just looking at the genes that they are clinically actionable, right? So so that allows us to get coverage for those dipintronics.
So what do you think? Do you think we should consider the third generation sequence, say the nanopore, get a longer sequence to identify this kind of splicing problem,
In research-wise, yes, those are definitely interesting approaches, and I think that's where the future is. Too, quantification is very important too, and it's something that I'm still trying to understand how nanopore PacBio can really quantify stuff. But it's the evolution, I think like genetic testing, we are in the infancy of this business, and DNA was first step, RNA and protein, all together. We need to have this integrated approach if we want to decrease VUS and improve the diagnostic yield.
So we have a question from Chrissy here.
Hi.
Hey, Chrissy.
Hello. I was wondering if there was a group of patients, based on personal history or family history that, would benefit more from this paired testing than, maybe some of the other patients would.
Yeah, interesting. It depends. For example, the ATM dipintronics, there was nothing special about them. Like you look at the cases, breast cancer, early breast cancer, like twins. Some genes don't, but others are very clinically striking, like the APC I showed you. And there are more examples, the MSH2, classic lynch we recently discovered too. Families that as clinicians, we look, what? This person is negative? How is it possible? You see segregation, you see everything, but you don't find the variant, you know? So we see both cases depending on the gene. So like our rationale to implement this clinic is like, you know, like when Myriad implemented MLPA, the L-DUP analysis, and that became standard, right? Because before we couldn't detect L-DUP analysis in BRCA1, BRCA2. And then in 2006, that was introduced, and then suddenly we were able to detect, and then that resulted in a 5% increase in the positive yield. And we now do in parallel, right? We don't do genetic testing and then we do MLPA, we do everything together. In RNA, we've seen like more updated number, 6 to 7% increase the positive yield, higher than they'll do. Just let's do that in parallel and detect more patients.
Right, thank you.
Good. Alice, so maybe I'll just send it back to you to wrap it up. I think we're done with the questions.
Okay, so actually I have one last question. So when you go back to the DNAs, do you only consider SNVs, or do you also consider other types of DNA variants as the cause of those splicing aberrations?
Oh, if they are associated, we look at everything, right? If you have a indel, or if you have a deletion of the last nucleotide, that happens sometimes, and things like that. I don't know. It depends on the variant, but we look everything pretty much.
Okay.
Yeah, for the splicing.
Yes. Okay. Thank you very much, everyone, and Dr. Karam for a wonderful talk. Yeah.
Thank you, guys. Have a good day. Stay safe there.
Yeah, thank you, everybody, and see you guys on Friday at 2 p.m. Bye.
Okay. Bye.